Good morning, everyone. Today we are going to study one of the most fascinating and the revolutionary topic in the pharmaceutics, that is liposomes. So before I begin, I want all of you to close your notebooks for a minute and imagine a real life situation. So imagine there is a patient suffering from a liver cancer and the doctor gives an anti-cancer injection through the patient's vein. So now I have a question. So where does the medicine go? Does it travels only the liver directly? Obviously no. So it travels everywhere. It reaches heart, brain, lungs, kidney, muscles, skin, bones, liver, cancer tissues. Okay, so the medicine has no intelligence. So it simply flows everywhere blood flows. Okay, so now think carefully. If the medicine is powerful enough to kill the cancer cells, so what happens when it reaches the healthy organs? So it also damages the healthy cells, no? So that is why the chemotherapy patients experience some hair loss, mouth ulcers, vomitings, loss of appetite, weakness, reduced immunity. So all these are all the side effects. Okay, so here I will give some real life example. So imagine a city. So a fire has broken out in only one building. So instead of sending one fire engine, the government floods the entire city. So will that solve the problem? No, no. So it creates even bigger problems. So exactly the same thing happens with the conventional medicines here. So instead of treating only the diseased tissues, they spread throughout the body. So scientists wanted a better solution here. So the big question, so can we design a medicine that knows where to go and protects itself during the travel, releases only at the disease site, reduces the side effects and improves the treatment? So the answer became the liposomes. So what is drug delivery? Students, many people think a pharmacy is only about making medicines, tablets like that. So actually the pharmacy is much more than that. So there are mainly three important questions. The first question is, which drug should we give? So that is the first question. And second question is, how much drug should we give? And how should we deliver the drug? So all these are the questions right now in the today's topic. Okay, so sometimes the success of the therapy depends more on how the drug is delivered than on the drug itself. Okay, so suppose if you have a very expensive smartphone, so will Amazon simply throw it outside your house? No, no. So first they pack it, the bubble wrap, foam, plastic cover, cardboard box, barcode, courier van, GPS tracking. Finally, the parcel reaches safely here. So now replace the smartphone with the drug, packing material with the liposome, courier van with the bloodstream, and house as a diseased cell. So now the entire concept becomes very easy now. So the liposomes are the protective drug carriers here. Okay, so like that, after entering the drug into the body as a dosage form like a liposome, it travels to the bloodstream and then it reaches the target cell, and finally the drug will be released there and it shows the therapeutic effect. Okay, so why conventional drug delivery fails? So let's understand why scientists developed these liposomes, and there are several reasons are there. The first problem with the conventional dosage form is the drug degradation. So many drugs are destroyed before reaching their target. Example, insulin. So if insulin tablets are swallowed, the digestive enzymes destroy them, and therefore insulin is given only by injections only. Okay, so there is no other option. The insulin should be given through injections. Okay, and second one is the poor solubility. So many new drugs do not dissolve properly in the water. So if there is a decrease in the solubility or a poor solubility, so what will happen? Automatically the drug absorption will also decreases. Okay, as a result of decreasing the absorption, so the bioavailability of the drug also will decreases. Okay, so hence if there is a poor solubility, there will be a poor absorption and poor bioavailability. Okay, example, paclitaxel, so a powerful anti-cancer drug which has a very poor soluble property, so liposomes improve its delivery. Okay, and next problem is the first pass metabolism. So one of the most important concepts it is, when we swallow the medicines, it travels mouth, stomach, small intestine, portal veins, liver, and finally it reaches the bloodstreams. So the liver metabolizes the many drugs here. This is called the first pass effect. Okay, again I'm repeating, the liver metabolizes many drugs. Okay, that effect or that phenomena is known as the first pass effect. Okay, sometimes more than 70% of the drug is destroyed before reaching the systemic circulation here. So the liposomes can help overcome this limitation in appropriate formulations and the route of administration also. Okay, and the next problem is the drug toxicity. Example, doxorubicin, excellent anti-cancer drug, but causes a heart damage. And amphotericin B, excellent antifungal drug, but causes a kidney toxicity. So scientists thought, instead of changing the drug, so why not change the carrier? So this simple idea changed the modern pharmacy. Okay, and what is NDDS first? So NDDS means novel drug delivery systems. So novel means a new, improved, advanced drug delivery system means a system designed to deliver the medicine safely, effectively, and to the appropriate site. So the liposomes are one of the earliest and the most successful examples of NDDS. Okay, so let us see the history of the liposomes first. So now let's discuss a little history here. So in the early 90s, British hematologist Elick D. Bangham was studying the phospholipids first. So when the phospholipids were mixed with water and examined under an electron microscope, they spontaneously formed tiny spherical vesicles here. So this discovery was remarkable because these vesicles closely resembles the membrane of the living cells. Okay, so the researchers soon realized that these structures could encapsulate the medicines and act as a drug carriers. So this marked the beginning of the liposome research. Okay, so which later leads to the clinically approved liposomal medicines later. Okay, so why are the liposomes are so special? So unlike many synthetic carriers, so liposomes are made from phospholipids. Okay, so which are the same molecules present in our cell membrane. No? So therefore our body generally accept them easily. So this property is called the biocompatibility. Okay, so as the property of the cell membrane in the humans are same as that of the medicine here, so there will be more chances for the compatibility to accept the foreign substance like medicine. So medicine is also a foreign substance here. Okay, so as the formulation is designed in such a way that will compatible with the cells present in our body, so this is called as biocompatibility. Okay, so let's see some small clinical example here. In case of patient A who receives medicine of conventional doxorubicin, having severe heart toxicity, hair loss, vomiting was seen. Okay, whereas in case of patient B receiving liposomal doxorubicin, so the drugs remain protected and higher concentration reaches the tumor tissues directly and reduce the cardiotoxicity, and the better patient tolerance is seen here. So this demonstrates how changing the carrier can improve the therapy without changing the active drug here. Okay, so imagine you are designing a medicine. So would you prefer medicine alone or medicine inside a protective carrier? Why? So think like a formulation scientist. So instead of simply memorizing the definitions. Okay, So once stop and just think. Okay, so before we proceed further, I want to ask one question. So have you ever seen a soap bubble floating in the air so many years? Yes. So now observe that bubble carefully. So it has a very thin outer membrane, empty space inside, a spherical shape. Okay, so the liposome is very similar to that. So except instead of air inside, it contains a water or medicine, and instead of soap molecules, it is made of phospholipids. That's it. Okay, so let's discuss the scientific definition here. So a liposome is a macroscopic spherical vesicle composed of one or more phospholipid bilayers enclosed in aqueous core. So this is the standard definition you will write in the examinations. So now let's understand every single word in this. Okay, so of course by hearting the definition is very important for your examination. Sometimes it will save you also. So many sequences in my life also I faced by hearting the definition. So many times it saved me. So definitely it will be give some clear cut and a clear picture about the topic for you guys. So by hearting is sometimes not sometimes, every time it will be helpful for you. So there will be so many situations are there in student life, obviously. So before going to the examination, sometimes you may forget some points. So by once you go through the definition, definitely it will give some information for you guys to write down points as your own. Okay, so sometimes there will be no time for you to revise all the topics at once in the examination itself. So at that time, if you just go through the definition simply, you can get the picture and you can able to write the points from that. Sometimes there will be no topic or no preparation at all before going to the examination. Some situations will be happen, obviously. So as a student we can't revise, or we can't go through all the topics at once at a short span of time. So sometimes we can skip, or we may skip some topics. So at that time, don't leave the topic, just go through the definition. I am telling that one. So here I am telling all these things. Why means don't leave the topic completely, just go through it or simply definition itself. Okay, so definitely it will be helpful one day. So I'm not telling about all the students. So some situations will be happen, obviously, for some time. Okay, so those who are ready to prepare or ready to go through the topic will be okay. So for others I'm telling this. Okay, if there is no time at all, means just focus on definition. Okay, definitely it will be helpful. So coming to the topic, now let's understand every single word in the definition here. Microscopic means it is too small to be seen with the naked eye. So the average diameter of a human hair is about 70 to 100 micrometers. So a typical liposomes may be 100 nanometers in the diameter here. Since 1 micrometer is equal to the 100 nanometers, a liposome is hundreds of times smaller than the width of the human hair. Will you imagine how thin the hair follicle is? So these liposomes are very smaller in size than the human hair. So to visualize this, imagine shrinking a football until it becomes a smaller than a bacterium. So that is the scale we discussing here. Okay, so why are the liposomes are spherical in shape only? So the nature often preferred the spherical shapes because they are energetically stable here. So spherical shape is more stable form compared to other forms. Okay, so examples includes soap bubbles, water droplets, air bubbles. So many biological vesicles. So a spherical encloses the maximum volume with the minimum surface area here. So this reduces the surface energy, making the structure more stable here. So this is why the phospholipid bilayers naturally curve and close into the spheres. Is it clear? Okay. Next, what are phospholipids means? So phospholipids are the amphiphilic molecules. Amphiphilic, break it down. Amphi means both and philic means loving. So an amphiphilic molecules has a hydrophilic head, that is water loving, and two hydrophobic fatty acid tails, that is water fearing. So the head contains in a phosphate group which is polar and interacts well with the water. Okay, and the tiles are long hydrocarbon chains, so which are non-polar in nature and avoid the water. So this dual nature is the key reason for the liposome formation. Okay, that is, so observe the diagram here. It has a hydrophilic head which is in spherical shape only, and the tile is hydrophobic one. Okay, so imagine this molecule as a person. The head loves to stand in water, and the tile hates water and always try to hide. So when thousands of these molecules are placed in water, what will happen? They arrange themselves automatically here. Okay, so everyday analogy, imagine people standing in a heavy rain. So everyone holds an umbrella above their head, so the umbrella faces the rain, so the person body stays protected under the umbrella here. Similarly, the hydrophilic heads faces the surrounding water, and the hydrophobic tile hides from the water. So this is a simple way to understand the phospholipid orientation here. Okay, self-assembly of the liposomes. So how do liposomes form? So now imagine adding phospholipids to water. So no scientist arranges them one by one. So instead they self-assemble. So this means they spontaneously organizes into a stable structure because of the chemical properties here. So the hydrophobic tails move inwards to avoid the water, and the hydrophilic heads remains in contact with the water. So a bilayer forms obviously here. So to eliminate the exposed edges, so the bilayer bends and seals itself into a closed sphere here. So this closed sphere is called the liposome. Okay, now let's discuss another important component, that is cholesterol. So many people think cholesterol is always harmful. So but in pharmaceutical formulations, cholesterol has a very important function. It stabilizes the liposome membrane here. So imagine constructing a brick wall. So bricks alone cannot form a strong wall here. So you need cement between the bricks. In liposomes, the phospholipids are the bricks and the cholesterol is like the cement. So it fits between the phospholipid molecules and reduce the membrane permeability, decreases the drug leakage, improves the rigidity, and enhances the stability during the storage. Okay. So if cholesterol is absent, the liposomes becomes more flexible and the drug may leak out during the storage and the shelf life decreases. So with an appropriate amount of cholesterol, the drug retention improves and the liposomes remains stable for longer. Okay. So let us discuss about this hydrophilic and hydrophobic drug loading. So this is one of the most frequently asked Viva Voice question also. So what are hydrophilic drugs? Means they dissolve in the water and therefore they are encapsulated in the aqueous core. Example, doxorubicin, cisplatins. And hydrophobic drugs, they mainly prefer the lipids and therefore they are incorporated into the phospholipid bilayer. Example, paclitaxel and amphotericin B. So water likes water, fat likes fat only. So students often remember this phrase during exams. Okay, so now I will ask some questions. Let's self check yourself whether you will give answers or not. Okay, why are phospholipids called the amphiphilic? So why do hydrophobic tails avoid water. So why do liposomes become spherical? So what is the role of cholesterol? Where will a hydrophilic drug be located? Where will be a hydrophobic drug be located? Okay, think once. Okay, if you know the answers, means just comment in the comment section. Classification of the liposomes. So liposomes are classified mainly based on the number of the phospholipid bilayers and the particle size. So there are mainly three basic types are there. First one is the small unilamellar vesicles, that is SUV. It has only one lipid bilayer and having a size of 20 to 100 nanometers, and these are having an excellent tissue penetration capabilities, and these are suitable for intravenous injections. Okay, and next one is the large unilamellar vesicles, that is LUV. So these are having one lipid bilayer only and having a size of 100 to 1000 nanometers. These are having very large aqueous core, okay, and which are having a capability to load. So these are used for higher loading of hydrophilic drugs. And third one is the multilamellar vesicles, that is MLV. So many phospholipid bilayers, they contains many phospholipid bilayers here. So look at the diagram. So look like an onion, okay. These are having a largest drug loading capacity and better sustained release action, okay. So here again I'm giving a small comparison, that is in case of SUV there is only one layer and having a size of very small, and drug capacity is very low here and stability is moderate, okay. Whereas in case of LUV, that is large unilamellar vesicles, uni itself indicates that it is single layer one, okay, and having a very large size compared to the SUV, and drug capacity is moderate here and the stability is very good, okay. And third one is MLV. In this it has many layers and the size is large only, and the capacity is very high and the stability is excellent, okay. So now pharmaceutical industries manufacture the modified liposomes also, that is sheath liposome, so covered with PEG. So its main purpose is to avoid the immune system recognition, remain in the blood circulation for a longer time, okay. So PEG coating acts like an invisible raincoat, okay. So the macrophages cannot easily recognize them, so the circulation time increases from hours to even days depending on the formulation. So clinical example for this is Doxil. So one of the world's most successful pegylated liposomal formulation here, that is PEG formulation, okay. Immunoliposomes, so the scientist attaches the antibodies to them. So these antibodies recognizes the cancer cells here, and the drug reaches only the cells expressing the target receptors. So very useful in the cancers, HER2 positive tumors, and monoclonal antibody researches also. So let us see some small clinical story here. So imagine a police searching for one criminal. There will be two situations, okay. So without photograph they search everybody, that is one condition, and in second condition with photograph immediately identify the criminal, okay. So antibody act like that photograph here, so they identify the correct deceased cell here. So next move on to the preparation methods. So now students, we know liposomes, so but how do industries manufacture millions of liposomes? So let's study preparation methods here. So there are many methods, first one is thin film hydration, reverse phase evaporation, ether injection, ethanol injection, detergent removal, sonication, extrusion. So among all these, the most important examination method is thin film hydration method, also called as Bangham method. thin film hydration method, this is also called as Bang-up method. So definition, so this is the most commonly used laboratory method for preparing the liposomes here. So principle here is the phospholipids and the cholesterol are dissolved in an organic solvent first. After solvent evaporation, a thin lipid film is formed. So the hydration of this film within an aqueous solution results in the spontaneous liposome formation here. So here, what are the steps? Means first dissolve the phospholipids and the cholesterol in a small chloroform or a menthol. So then transfer the solution into a round bottomed flask and remove the solvent using a rotary evaporator. So a thin lipid film forms on the flask walls here. So after phosphate buffers or distilled water, and then add the phosphate buffer or the distilled water, the lipid film hydrates and forms the multilamellar liposomes here. So reduce particle size by sonication or extrusion methods. Okay, advantages of this method is very simple and a reliable one, and it is suitable for many drugs and produces a stable liposomes. Okay, and disadvantages of this method are uses the organic solvents. Initial liposomes are large and requires a size reduction here. And second one is reverse phase evaporation method. So what's the principle involved here is a water in oil emulsion is prepared and removal of the organic solvent produces the liposomes here. So lipids are dissolved in the organic solvent and the aqueous drug solution is added to the formulation and the water in oil emulsion is formed. So this emulsion is sonicated for uniform mixing here. So the organic solvent is evaporated under reduced pressures. The large unilamellar liposomes with high drug entrapments are formed here. Okay, this is how we will prepare the liposomes by using a reverse phase evaporation. And the next method is ethanol injection method. So the lipids dissolved in the ethanol are rapidly injected into an aqueous phase, causing a spontaneous liposome formation here. So that is the basic principle involved. And here what are the steps means, first dissolve the lipids in ethanol, inject into a warm buffer while stirring, and then ethanol diffuses into the water and the liposomes form immediately here. So remove the ethanol if required. Okay, this is how we will get the liposomes. It is a very simple method and easy to scale up. Okay, no rotary evaporator required here. Okay, and next method is ether injection method. So in this method, what's the principle involved means, lipids first dissolved in the diethyl ether are injected into the heated aqueous phases. So and then ether evaporates and leaving the liposomes here. So advantages of this method is it produces unilamellar vesicles and a very simple process. But the disadvantages are ether is highly flammable and residual solvents may remain here. Okay, and the next one is detergent removal method. So lipids are mixed with the detergents to form the mixed micelles here. So and then removal of the detergent allows the liposomes to form. That is the basic principle. So the method of the detergent removal may be dialysis, gel filtration or adsorption. Okay, so advantages of this method are it is good for the membrane proteins and the uniform liposomes. And the next one is the sonication method. So what's the principle involved means, so ultrasonic energy breaks the large liposomes into the smaller liposomes. So what's the procedure adapted means, first prepare the unilamellar liposomes and apply the ultrasonic waves, and the large vesicles breaks into a smaller unilamellar vesicles. Okay, this is how we will prepare liposomes. Okay, so advantages of this method is it mainly produces the uniform small liposomes and improves the particle size distribution also. Okay, extrusion method. So what's the principle involved means, so liposomes are forced through the membrane with a definite pore size to obtain the uniform particles here. Okay, what are the steps means, first prepare the liposome suspension and then passes through the polycarbonate membrane and repeat several times. Uniform size liposomes are obtained by this method. Okay, so advantages means it having a narrow particle size distribution and reproducible results are there. So it's suitable for the industrial production only. Okay, So let's compare all the methods. So this thin film hydration method is most common method. In case of reverse phase evaporation, what's the main feature? Means it's having a high drug loading capability. Ethanol injection method, it is a very rapid preparation method, and ether injection, it is suitable for produce unilamellar vesicles. Okay, and detergent removal method. So the membrane protein studies can be done by this method. Sonication, so size reduction can be achieved here by this method, and extrusion, so uniform particle size can be achieved by this method. Okay, so all these are the main features of all the methods. Okay, so industrial manufacturing of liposomes. So industrial manufacturing of liposomes is carried out to produce the large quantities of the high quality liposomal formulations here, and the pharmaceutical grade phospholipids, cholesterol, drug, and the purified water are selected as the raw materials here, and the lipids are dissolved in the suitable organic solvents to prepare a homogeneous lipid solution, and liposomes are formed using methods such as thin film hydration, ethanol injections, or high pressure homogenization. So the lipid solution is hydrated with an aqueous phase to form the liposomal vesicle here. So the drug is encapsulated either in the aqueous core or within the lipid bilayer depending on the properties. We will prefer the method, and the large liposomes are reduced to the desired size using in a sonication or extrusion or microfluidization. Okay, so the liposomal suspension is processed under aseptic conditions to maintain the sterility, and the sterile formulation is performed whenever suitable to remove the microbial contamination, so while preserving the product quality here. So every batch undergoes quality control tests including particle size, zeta potential, and the entrapment efficiency. So the additional tests such as sterility, pH, drug content, endotoxin levels, and the stability are also performed in industries. Okay, so only batches that meet all the specifications are approved for production release. So the finished liposomal formulation is aseptically filled into the sterile vials or ampules and sealed. Okay, so the final product is labeled, packed, and stored under recommended storage conditions before distribution. So the industrial manufacturing ensures mainly that the liposomal medicines are safe, sterile, stable, effective, and of constant quality for patient use. Okay, characterization of the liposomes. Students, imagine you are constructing a bridge. So after construction, can we immediately allow vehicles to use it? No. First, engineers check the strength, thickness, stability, safety, load bearing capacity. So similarly, after preparing liposomes, the pharmaceutical scientist must evaluate their quality. So this process is called the characterization. So without characterization, we cannot know whether the liposomes are suitable for patient use. Okay, so suppose a company prepares a liposomal formulation today. So questions arises are, so are all liposomes the same size? Is the drug actually entrapped in or encapsulated? So will the drug leak during the storage? So will the liposomes remain stable for 6 months? So are they sterile? So characterization answers all these questions. The first parameter is particle size. The particle size is one of the most important characteristics. So why? Because the particle size determines the blood circulation time, drug release, cellular uptake, and the biodistribution clearance by the macrophages. So clinical example here is, suppose a liposome is 80 nanometers. It's easily circulated through the blood vessels. So now suppose another liposome is there having the size of 5 micrometers. Can it passes through the small capillaries? No. So it may because entrapped or removed rapidly by the immune system. So therefore a controlled particle size is extremely important here. Okay, instrument used. So dynamic light scattering, that is DLC. Example, laser light hits particles, the particles scatter lights. So computer calculates the average particle diameter here. So why should intravenous liposomes be small? Example, the expected answer will be to improve the circulation and avoid the blockage of the small blood vessels here. Okay, and the second parameter is zeta potential. So now let's study another important parameter. So many students find this difficult, so let's simplify it. Okay, so imagine two magnets, north, do they attract? No. So the repel, exactly. So liposomes also possesses electrical charges, so this surface charge is called the zeta potential here. So why is zeta potential important? So if liposomes have a very little surface charges, they tends to aggregate. So the aggregation means sticking together, so large aggregates becomes unstable here. So if the zeta potential is sufficiently positive or negative, so the particles repel one another and keeping the suspension stable here. Okay, example, imagine 100 school students. So if everyone stands very close together, they collide. So if everyone maintains some distance, movement becomes very easier here. Similarly, so electrical repulsion keeps the liposome separated. And the third parameter is entrapment efficiency. So this is one of the most frequently asked Viva Voice question. So not all of the drug added during preparation becomes encapsulated, some remains free in the solution. Therefore, we calculate the entrapment efficiency here. So entrapment efficiency, it is calculated in percentages, is equal to entrapped drug by total drug added into 100. So with this formula we can calculate the entrapment efficiency, that is EE. So I will give some numerical example, like if there is a total drug added is 100 mg and the drug encapsulated is 80 mg, so EE will be 80 by 100 into 100, that is equal to 80%. So this means 80% of the drug is successfully loaded into the liposomes here. So why is high entrapment efficiency very important? Because less drug is wasted, lower production cost, better therapeutic effect, and the more efficient formulation can be designed. Okay, so because of all these parameters, so entrapment efficiency is very important. And morphology, so scientists also examine the shape, lamellarity, surface characteristics also using transmission electron microscope, that is TEM, and cryo-electron microscope, cryo-EM. So these techniques allow direct visualization of the liposome structure here. Drug loading. So now let's understand how drugs are loaded into the liposomes. First is hydrophilic drugs. So water soluble drugs dissolves in the aqueous core. Example, doxorubicin, cisplatin. Hydrophobic drugs. So lipid soluble drugs insert into the phospholipid bilayer here. Example, paclitaxels, amphotericin B. So amphiphilic drugs. Some drugs have both hydrophilic and hydrophobic regions. So these may partition between the aqueous core and the lipid bilayer depending on their physiochemical properties. So take a glass of water, a spoon of cooking oil, add food color to it, it mixes with water. Add a drop of oil, it remains separate. Example, water soluble drugs behaves like the food color here, and the lipid soluble drugs behave like the oil here. So this simple demonstration helps students visualize the drug loading here. Okay, drug release mechanism. So students, after a liposome reaches the target tissue, how does the drug enters the cell? So there are several mechanisms are there. First one is fusion. So the liposome membrane merges with the cell membrane, so the drug is released directly into the cytoplasm here. And second is endocytosis. The cell membrane surrounds the liposome, and the liposome is internalized into the cell. So later the drug is released inside the intracellular compartments. And third one is adsorption. So the liposome attaches to the cell surface. So the drug molecules then diffuses into the cell. So that is the another. And fourth one is lipid exchange. So sometimes phospholipids from the liposome exchanges with the phospholipids in the cell membrane. So this can facilitate the drug transfer here. Passive targeting. So cancer blood vessels are abnormal, so they contain the large gaps, so the liposomes accumulates more readily in these leaky blood vessels. So this phenomena is known as the enhanced permeability and the retention effect. So the passive targeting does not require any specific ligand or antibody here. And active targeting. So scientists can attach antibodies, peptides, sugars, vitamins to the surface of the liposomes here. So these ligands recognizes the specific receptors on the target cell. Example, HER2 targeted liposomes for HER2 positive breast cancers. And a clinical case. So patient here receives a conventional anti-cancer drug. The drug distributes throughout the body here. Healthy tissues are affected. But whereas in case of patient B receives a targeted liposomal formulation, experiences a greater proportion of the drug reaches the tumor here, and the side effects are reduced. Okay. So here I am recapping again the entire process. In the first stage, the drug is loaded inside the liposome. So after administration, the liposomes reaches the bloodstreams, and from the bloodstreams it reached to the tumor cell at the target site. Okay, so in target site it may adopt any method like fusion, endocytosis, adsorption. So whatever may be the method, it releases the drug. Okay, and thereby it produces the therapeutic action. Okay, so this is the entire sequence. So like this you may get questions in your exams like why is particle size important, what is zeta potential, define the entrapment efficiency, differentiate the hydrophilic and the hydrophobic drug loading, explain fusion, explain endocytosis, what is passive targeting, what is active targeting, so explain the EPR effect. So like that you will get questions. So in GPAT and NIPER point of view, dynamic light scattering, that is DLC, the particle size analysis is very important. TEM, cryo-UM, that is morphology. Okay, and high zeta potential, that is better colloidal stability is also important. Entrapment efficiency is a key formulation parameter, and EPR effect is the basis of the passive tumor targeting. So like this we have to focus. Okay, applications of the liposomes. Students, now that we know how liposomes are prepared and how they deliver the drug, and the next question is, where are liposomes used? So the answer is almost every major area of the modern medicine here. So the liposomes have transformed how medicines are delivered because they can carry the drugs more safely and effectively here. In case of cancer therapy, that is the first example. So cancer treatment is the largest application of liposomes. So normally chemotherapy drugs circulates throughout the body and damages the healthy cells. So the liposomes help by protecting the drug in circulation, increases the accumulation in the tumors through the EPR effect, and reducing the toxicity to healthy organs. Example, doxil, that is liposomal doxorubicin. Benefits for this is reduced cardiotoxicity, longer circulation time, better tumor targeting. And second application is antifungal therapy. Example is amphotericin B. So conventional amphotericin B is highly effective but causes significant nephrotoxicities. So liposomal formulation, that is ambisome. So advantages of this is reduced kidney toxicity, higher tolerated doses, better treatment of systemic fungal infections. And third application is vaccines. So liposomes can deliver the protein antigens, peptides, nucleic acids also. So they improve the immune responses by facilitating uptake by antigen presenting cells in gene therapy. Well, this is the next application. So DNA and RNA are unstable and easily degraded. So liposomes protects these molecules and facilitates the cellular uptake here. Applications includes gene replacement, gene editing, RNA therapeutics. And fifth application is ophthalmic drug delivery. So eye drops often remains on the eye surface only briefly. So liposomes increases the resident time and improves the corneal penetration and reduces the dosing frequency also. And the sixth is cosmetic industry. So in cosmetic industry also liposomes are incorporated as anti-aging creams, vitamin formulations, moisturizers. So they enhances the penetration into the superficial skin layers. In brain drug delivery also. So the blood-brain barrier prevents many drugs from entering the brain. So researchers have developed specialized liposomes to improve the drug delivery for Alzheimer's disease, Parkinson's disease, and the brain tumors also. Advantages of the liposomes. So every pharmaceutical technology has a strength. So let's summarize why liposomes become so successful here. First advantage is targeted drug delivery. So the drug concentrates more at the desired site. Okay, that is the main advantages of this liposomes. And second one is reduced toxicity. So healthy tissues receives a lower drug exposure. Okay, and third is improved bioavailability. So more drug reaches the systemic circulation or the target tissue. With this we can achieve the more bioavailability. And the sustained drug release. So liposomes can release the drug gradually over time. Okay. Fifth, protection from the degradation. So the liposomes protects the drug against the enzymes, hydrolysis, oxidation. And the next advantage is ability to carry the different types of drugs. Hydrophilic drugs, aqueous core, hydrophobic drugs, lipid bilayer, like that. Okay. And seventh one is biocompatibility. So because these phospholipids resembles the cell membranes, so liposomes are generally well tolerated and having a high biocompatibility. Disadvantages. So although liposomes are excellent carriers, they are not perfect. There are some limitations are there, that is high manufacturing cost, limited storage stability, drug leakage during storage, oxidation of the phospholipids, scale up challenges, sterility requirements. So if liposomes are so effective, why are not all medicines formulated as liposomes? So expected answer is cost, manufacturing complexity, stability, limitations. So all these are all the various limitations of these liposomes. So marketed products. So let's discuss the medicines you may encounter in hospitals. That is Doxil drug, doxorubicin, used for the breast cancer, ovarian cancer, and Kaposi sarcoma. And second one is Ambisome drug. Here is amphotericin B, used for several fungal infections. Myoset drug here is doxorubicin. It is also for breast cancers. And the future scope is liposomes research is rapidly expanded into personalized medicine, targeted cancer immunotherapy, gene editing, mRNA therapeutics, antifungal, intelligence assisted formulation design. Okay, so the final revision. So let's summarize everything we are learned. Okay, first is definition. So liposomes are the microscopic phospholipid vesicles enclosed in an aqueous core. Components here are the phospholipids, cholesterol, drug, and water. And the types are SUV, LUV, MLV, pegylated liposomes, and immunoliposomes. Preparation methods are thin film hydration, reverse phase evaporation, ethanol injection, sonication, and extrusion. Characteristics: particle size, jitter potential, entrapment efficiency, and morphology. Drug loading: hydrophilic core, hydrophobic bilayer. Targeting: passive targeting, active targeting are there. So passive targeting means EPR effect, and active targeting means ligand or antibody mediated one. Applications are in cancer, antifungal therapies, vaccines, gene therapy, ophthalmic formulations, cosmetics, and the brain drug delivery. So always remember this: a medicine can only work effectively if it reaches the right place at the right concentration and at the right time. Okay, so the liposomes help achieve this goal by acting as an intelligent drug carrier here. So they protect the medicine, improve the delivery, reduces the side effects, and represents one of the greatest advances in the modern pharmaceutics here. So as a future pharmacist and a pharmaceutical scientist, so your responsibility is not only to know the drug but also to understand how to deliver it safely and effectively to improve the patient lives. So thank you and wish you success in your studies and the future.